आएगा ना लिंक, ये वाला तो नहीं होगा ना यूज?
Miss Hanfa, your voice is not clear. Somehow distortion at your backside.
ओके, मैं देखती हूँ, ठीक है। अब अब ठीक है? ये सबसे अब ठीक है?
Yes, now it's clear. Yes, it's clear.
ओके।
Like ऐसे लग रहा है जैसे आपकी बैक साइड पे कोई शोर वगैरह है, तो उसकी थोड़ी सी डिस्टॉर्शन हो
वो
रही है।
वो फैन की थी।
और उसी वजह से वो बस वो इसी की लग रहा है, इसलिए आपकी वॉइस थोड़ी लो हो रही है, लेकिन अभी ठीक है, कंटिन्यू हो सकता है।
ओके, जब स्टार्ट करना हो आप मुझे बता दीजिए, तो मैं तब स्टार्ट कर दूँगी, ठीक?
Yes, I will start at 8 and after giving your intro, I will invite you.
ओके।
Miss Hanfa,
जी, जी,
मुझे
आवाज़ आ
अपना
रही है?
कंप्लीट नेम बताइए।
हल्फा ईमान।
ईमान।
जी,
ओके।
जी, ठीक है।
Assalamualaikum and a very warm welcome to everyone for joining us today. Here is Hafsa Sarfraz. Currently, I am pursuing my MSc Honors Plant Breeding and Genetics from University of Agriculture, Faisalabad. Besides my educational career, I have the honor to serve as the General Secretary at VitaNova International Alliance for Sciences. It brings me immense joy to welcome you all to the fourth official session of VNIAS Biotechnology and Bioinformatics Summer Internship. Your enthusiasm and commitment to learning are truly appreciated. Now, I would like to introduce our today's class instructor, Miss Hanfa Imam. She is serving as the researcher at VitaNova International Alliance for Sciences. Besides this, about her professional career, she is a research scholar at Center of Biotechnology, Agriculture, Biochemistry, and Biotechnology. Before we start our today's session, please make sure your availability in the session, and if you guys have any queries, you can write and ask at the end. Now, I would like to give a warm welcome to our today's instructor, Miss Hanfa. You can start now. The floor is yours.
Thank you, Miss. Assalamualaikum. I am excited to welcome you this session under the VINAS Summer Internship Program 2020-25. Today's presentation focuses on bioinformatics. Our goal is to understand its origin, roles, tools, and application in the sector like agriculture, medicine, and modern research. So let's start our topic. What is bioinformatics? Next slide. Bioinformatics is derived from two words, bio meaning life and informatics which refer to the science of information processing. So bioinformatics is literally the science of processing biology information. It is the field combining biology, computer science, mathematics, and statistics to analyze biological data. At its core, bioinformatics aims to help researchers clinically make sense of massive biological datasets such as DNA sequences, protein structures, and gene expression patterns. These datasets are too complicated for manual analysis, and that's why software tools and databases come in. So our focus in collection, gathering data through sequencing techniques, lab expertise, and databases. Storage, using structured databases to store genomic, proteomics, and metabolomics data. So its analysis, applying algorithms to understand function, mutation, expression, or evolutionary relationships. Finding Share findings through platforms like NCBI, Uniport, or researchers worldwide can access it. Bioinformatics is essential in the fields like genomics, proteomics, metabolomics, and system biology. It also powers tools like BLAST, sequence alignment, and GeneBank like data storage. Next slide, please. Next slide.
Miss, you can do it by yourself to move the slides next.
Okay. Here is issue. It can't. Next. Next. History of bioinformatics. The seeds of bioinformatics were planted in 1960, where scientists first attempted to understand protein and DNA sequences using computers. This early bioinformatics was manual, slow, and limited. In 1980s and 90s, the field exploded with launch of Human Genome Project. The goal of Human Genome Project was to sequence all 3 million basis pair of human genome. It took 13 years. It produced vast data which needed advanced tools to analyze and store. In this modern era, with the rise of next-generation sequencing, we can sequence entire human genome sequence in hours, along with AI, artificial intelligence, machine learning, and cloud community. Bioinformatics now play a central role in drug development, disease diagnosis, precision medicine, and agriculture. BLAST was developed in 1990 to quickly compare gene sequence. The first biological databases like GeneBank appeared in 1980s. This field has grown due to large technological advances and biological demands. Next slide. This slide likely contains a timeline or visual summary of what we can discuss. Let's talk through the milestone. In the left side, manual data entry and early protein structure discovered. In the middle, it shows that Human Genome Project 13-year project in which human genome sequence developed. On the right side, in this picture show modern bioinformatics with NGS, AI, and global databases. From the manual data entry to the AI-driven analysis, bioinformatics has become a pillar of modern biology. Next slide. Bioinformatics in agriculture. Gene discovery from the crop development. Gene discovery bioinformatics help us identify genes responsible for traits like drought resistance, early ripening, or high yields. We can use genome sequencing or bioinformatics tools to locate and edit these genes. Pest resistance. Pest and disease resistant research. Using comparative genomics, we can find natural resistance genes and breed crops that are immune to common pests and pathogens. Third is pest precision agriculture. With the genomic data, we can guide farmers on which crop to plant based on climate and soil. Soil analysis. We use microbial DNA in soil to understand its health and fertility using tools like metagenomic analysis. For example, golden rice bioinformatics help us insert vitamin A producing gene, BT crop carrier pest resistance gene discovered using bioinformatics. Next slide. Bioinformatics in medicine. Its application in medicine, personalized medicine, and pharmacogenomics. Revolution in medicine. Bioinformatics has completely transformed the field of medicine. We are no longer treating disease in one-size-fits-all way. Instead, we can now understand how a person's gene influences their responsible to drugs or their likelihood of developing a disease. Personalized medicine, thanks to bioinformatics, and doctor can analyze a patient's DNA or gene expression or choose treatments that are mostly like to work. This is called personalized or precision medicine. Disease gene identification. We can identify genes responsible for disease like Alzheimer or BRCA1 and 2 in breast cancer by comparing sequenced using databases like NCBI or many more. Cancer genomics. Cancer genomics and diagnostics. Cancer is often caused by genetic mutation. Bioinformatics allow us to sequence tumor, identify these mutations, and even match patients to most effective therapies or clinical trials. Drug target discovery and vaccine development. Drug target discovery. Bioinformatic tools like molecular docking or PDB protein structure analysis help a scientist find where a drug might help on a protein to stop disease process. And vaccination like COVID-19 and messenger RNA used in vaccines were created quickly using bioinformatics to the model the virus's spike protein and stimulate the immune response. Next slide. In the next slide, we will discuss bioinformatics in daily life. What the role of bioinformatics in our daily life? Bioinformatics is not just for scientists, it's also the part of everyday life. Like genetic testing, many of you have heard like DNA. These use bioinformatics to decode your DNA and tell us about your heredity or health needs. Wellness or nutrition apps. These apps are used that analyze your genome and suggest the best food, supplements, or exercise routines to your DNA. Next, agriculture productive through the informed practices. Informed agriculture. Farmers use genome-based software to choose optimal seed, manage soil nutrients, and maximum crop yield based on bioinformatics prediction. This picture also shows that the role of bioinformatics in our daily life. Next slide. What is databases? But databases in bioinformatics is like a digital library. It stores biological data in structured and searchable way. Data like data type like includes DNA sequences like ATGC codes, protein sequences and structures, gene functions, mutations, 3D model, and pathway diagrams. So these databases allow researchers to retrieve information and compare genes between species and find links to disease or a traits. If a scientist sequences a new gene, they compare it to existing entire in gene bank or Uniport to see what it might do. Next slide. The role of databases in bioinformatics. Bioinformatics databases are the backbone of modern biology. Global storage hub, they store massive amount of data from researcher lab, genome project, hospital, etc. It also enables tasks like sequence alignments, finding matches, mismatches, mutations in the sequences, and also help us gene annotation like labeling function or position of the protein or a gene and it's comparing across the species. Most databases like NCBI example are free and open resources used by a researcher worldwide. Common bioinformatics databases like NCBI, Uniport, that PDB, CAC, example DDBJ, EMBL, and GeneBank. Next slide. In this slide, we will discuss about Common bioinformatics databases like NCBI, National Center for Biotechnology Information. It is used for hub for gene, protein, nucleotide sequence tools like BLAST and research articles through it. PubMed, Uniport, protein sequences and functions. Compress- Comprehensive protein sequences and function databases used for study domains, interactions, and their pathways. PDB, protein structure data, the protein data bank. It holds 3D structures used in drug discovery to visualize how drugs to bind to protein. Tech focuses on pathway and their functions, help map gene to metabolic pathways. And a genome browser let us use the annotated genome from human, mice, plant, different species, etc. GeneBank, uh, DDBJ, it's also a common databases in bioinformatics. Global that share nucleotide sequence often for consistency. Next slide, please. Using NCBI how to research. First, we discuss about NCBI database. Go to this website NCBI and choose a database. This is the homepage of NCBI. In the next slide, we will show the homepage of NCBI. It connects over 40 different databases including genes, PubMed, protein, genome, structure, and many more. First, choose a database, nucleotide, or a protein or gene. Enter a query like BRCA1 Homo sapien. It's a breast cancer gene derived from the human gene. Uh, Select the type of biological data you want. For example, use gene to search for specific gene, use protein for amino acid sequence, use nucleotide for DNA and RNA, use PubMed to find scientific articles. BRCA1 Homo sapiens, this will open the gene bank for the human BRCA1 gene linked to breast cancer. You will see the gene summary, function, and chromosomal location, sequence data, DNA and protein, associated conditions like breast cancer, research articles from this database. It may also use for BLAST, paste any DNA or protein sequence to find matches or across the species. PubMed also explored the latest research on this page. Next slide. Like this is an example how to search in NCBI. TP53 gene Homo sapien, search in NCBI database and results show the gene function, its location, its sequence and diseases. Also link data with gene bank, protein translation and its publication. Next slide. This is the overview of how to search using NCBI. Like in the left side, you see the gene I selected, and in search bar I enter the Homo sapien gene, and then search it will show the gene, its structure, its function, and its related publication. Next slide. Uniport. In Uniport search, first of all, P53 human protein and its show protein function, its domain and its structure. In the next slide, it will show amino it will show amino acid sequence in its variant function act as transcription factor and tumor suppressor gene. Interact with M2 DNA. Domains is domain like trans-activation domain, DNA binding domain, and its pathway use apoptosis cell cycle arrest. If study cancer, this data helps design drugs to activate or stabilize P53 in tumor. Next slide. Let's show the Uniport how we search about it. Yeah, next slide. PDB. PDB 1 TUP P53 protein structure. It will show 3D view of DNA binding protein of P53 where mutation like R22480Q occurs. How small molecule might bind. It used for the structure-based drug designing. View binding pockets, run docking simulations, predict side effects. Next slide. This is the conclusion slide. Importance of databases. Databases are the backbone of bioinformatics research. They enable fast and global and accurate analysis. Mastering them is essential for any career in bioinformatics for our daily life, like efficient management, prevent large data, quick retrieval. What we learn from these databases like DNA, RNA, protein sequence, mutations that may cause disease, structural changes linked to drug response, gene mutations, and their functions in role in their pathway, and also help us literature references for every data point. We use them to make the sense of complex biological data to reduce lab cost and time by predicting using data in silico to share findings and build knowledge globally. It gives us faster diagnosis of genetic diseases, more targeted drug discovery, insight into evolution and development, better crop and animal genetics in agriculture. Bioinformatic tools and databases allow us to explore biology at the molecular level, generate new discoveries, and contribute to fields as diverse as medicine, agriculture, and biotechnology. Next slide. This is our internship program in which we discuss about hands-on training in bioinformatics tools and workshop on data analysis and its interpretations. In the next class, we will discuss about the bioinformatics tools, BLAS, multiple sequence alignment, and different tools. We will discuss about it in next classes. Okay, next. Okay, thank you, Max. Any question?
Thank you so much, Miss Hanfa, for such a informative session. If you guys have any question, you can ask in the chat box, and I will, Miss Hanfa will answer the questions. Please write in the chat box. Here is a question from Burhan Heather. What are genome assemblies? Can you please provide an insight about it?
Basically, we will discuss in next class all about this. Like last, we will discuss throughout.
Rimsha
Uh,
Fatma, you have a question about the
yeah, thank you so much, everyone, to join us in today's lecture. First of all, I would like to answer the question of Mr. Burhan. Burhan, basically, when we sequence the data, like as I already told you, we get data through DNA extraction, molecular analysis, and through blotting process. Then we go towards sequencing through different sequencing techniques. Now, for third and fourth generation sequencing techniques are working. After getting data from sequencing, we move forward with bioinformatics tools, okay, uh, to analyze it more precisely. ठीक है? So that's data that we get from sequencing uh, genome data, genome assemblies. Okay. Next, Miss Rimisha question. Miss Afsa, please elaborate.
She basically has a question about the core concept of blast and when we perform blast.
Yes, Miss Hanfa, please go ahead.
Again, question.
The question is that how we use blast and when we need to use blast. That is blast tool.
Okay. Last is basically local alignment search tool. It is a powerful algorithm used to compare DNA query or protein sequence against databases to find out similarities and differences between the species. We use it to new DNA sequence from our experiment and want to know what gene it belongs to. Like we isolate a gene from a plant and we paste it in a sequence in BLAST. It matches a chlorophyll or biosynthesis in gene in rice. Also check we use BRCA1 gene in human is similar to the same gene in chimpanzee. Like we use to identify similarities between the species.
Anyone have any question? You can ask. And if anyone want to share your answer in as a host, you can share your answer. I will share you a link of hosting here in chat box. Okay. So
Okay.
yes, if anyone has any question and want to be a host as a questioner,
Here is a question from Mr. Mughal. What does cloud computing plays role in bioinformatics?
Okay, basically, cloud computing, we use cloud computing for large database bases. Like when we have too much large genome base, database, for example, human genome project, that human project data data was too much large. To deal with that type of data, we use cloud computing to manage a large and vast type of data. That in this case we use cloud computing in bioinformatics. Yes, obviously the bioinformatics is a vast domain. It's have like applications in all life sciences domain. I mean to say that in biology domain, you can take agriculture, you can take medicine, you can take any where biological system involves. Their bioinformatics involved to analyze biological data.
Okay, thank you, sir. Here is the next question. What is PDB and how to use it? Miss Anfa? Miss Anfa, is I audible to you?
I think Miss Hanfa is not here due to some issues.
Okay.
Yep. If I talk about PDB, PDB is a database as she already mentioned. Database used to uh, check the previous data about genes. Okay, just like a gene bank. Okay.
Okay, sir, got it. Miss Anfa is joining within 2 minutes. Then we can move forward.
Uh, there's no challenges in bioinformatics, but its major issue in bioinformatics. We we have mean to say that we have best computer and best databases. Uh, mean to say that it's much expensive. Uh, we need supercomputers to deal with large databases, uh, just like Human Genome Project and other any other large project. We need very strong, you can say, databases and computer, and like just like supercomputers. Okay, to analyze big large size of data. Yeah, that's it. That issue is always in bioinformatics, as most of the institutions they have not more fundings. So for purchasing new and high quality databases, just like Mr. Was telling about uh, cloud computing. Basically, scientists use mean to say that the people have not facility to access the large data, so they use cloud computing as an alternative to deal with uh, large size of biological data. But the reason is that cloud computing is not safe. Mean to say that there is issue of leakage of data in cloud computing because you have not direct access. Yes. If I talk about bioinformatics, scientists called, you can say scientists called bioinformatics as gold biotechnology. Okay, gold biotechnology. You can imagine it. It's very important. It's a application in all field, even in microbiology, in biotechnology, in in other domains like biochemistry, in zoology, in animal data, in biologic. Mean to say that where the biological system involves, there obviously, yeah, there obviously bioinformatics involved. Mean the biological system, or you can say biological fields cannot survive now without bioinformatics. That's why it's called gold biotechnology. As last time, like Miss Minza was telling that if any microbiology opportunity is here, then we can inform her. The point is that basically, if we see the modern definition of biotechnology, it's the applied use of biotechnology. It's a applied use of, you can say, bioinformatics, microbiology, biochemistry. Okay, mean to say that combine these all domains, and it's convert into biotechnology. Biotechnology is not not a biotechnology without biochemistry, microbiology, bioinformatics. So biotechnology is just not a field, it's too much applied field, and
Everyone, please wait. There is a connectivity issue. We will resolve it soon.
Sorry for inconvenience.